Hello again. We continue in our document 1 immunodeficiency, and we arrive to the second class of immunodeficiencies, which is the secondary or acquired immunodeficiencies. What are the causes of the acquired immunodeficiencies? There are many factors that contribute or may contribute to acquired immunodeficiencies. We list the malnutrition, having bad lifestyle or bad diet. Excuse me. Cancer also may lead to acquired immunodeficiencies, since in cancer we have multiplications, fast multiplication, rapid multiplication of cancer cells. So the immune system becomes weak, and the cytotoxic T lymphocytes also they cannot kill or destroy all cancer cells. We have depletion in immune cells. Also, the immunosuppressive treatments or drugs, in the case of organ transplants, the receiver of the organ transplant receives immunosuppressive drugs in order to uh, decrease or to eliminate the immune responses triggered by the receiver to attack organ transplants in the cases where we have incompatibility in MHC molecules between the donor and the receiver. So the immunosuppressive drugs also leads to immunodeficiencies, mainly in the receiver. The chemotherapy and the radiotherapy for cancer, as you know, chemotherapy kills all stem cells in active or highly active mitosis, including the stem cells of the bone marrow. Also, the radiotherapy kills both cancer and normal cells. And the main cause of acquired immunodeficiencies is the human immunodeficiency virus, HIV, that causes the acquired immunodeficiency syndrome or AIDS. So in our video, we are going to explain the mode of action, the mode of transmission of HIV, its structure, and the progression of the infection with HIV. How does HIV lead to acquired immunodeficiency? So first, let's study the HIV structure. HIV is a retrovirus. Retro means backward. This means that in order to reproduce, the HIV transcribes its RNA into DNA. So the genome of the HIV is made up of RNA molecules instead of DNA. So in order to reproduce, HIV should transcribe its RNA into DNA. That's why it is called retrovirus. And to transcribe the RNA into DNA, it uses its enzyme called reverse transcriptase. It is not a RNA transcriptase, but a reverse transcriptase that transcribes the RNA into DNA. And once the DNA is formed, the viral DNA is formed, it will be inserted into the genome of the infected host cell. So in this way, the HIV virus multiplies within the host cell by transcribing its RNA into DNA, then by inserting its DNA into the DNA of the host infected cell. So the HIV has two identical RNase molecules in blue here, the blue chains, and some proteins. So the RNA genome contains identical RNA molecules and proteins called protease, integrase in yellow, and a reverse transcriptase in red. This genome, the set of RNAs and associated proteins, is contained in a protein capsid here in light brown. The capsid is an envelope that protects the viral genome. The protein capsid itself surrounded by a lipid envelope of glycoproteins in purple here. The glycoproteins expressed on the surface of the virus are called the viral proteins or the viral antigens. And these envelope proteins or envelope antigens will be recognized by antibodies or by our immune system. The glycoproteins are mainly, we list the glycoprotein 41, which is a transmembrane glycoprotein, and we have the glycoprotein 120. These glycoproteins will allow the binding of the virus to its host cell and its fusion with the host cell membrane in order to release its content, its genome, in the cytoplasm of the host cell. So the structure of HIV should be studied and memorized. It is a retrovirus. It contains RNA instead of DNA. It transcribes its RNA into DNA using reverse transcriptase. Once the RNA is transcribed, the obtained DNA, viral DNA, will be inserted into the genome of the infected host cell, and in this way the HIV multiplies. The envelope of the HIV virus contains or presents glycoproteins. The glycoproteins are called viral antigens that are recognized by the immune system. To the modes of transmission of HIV, we have sexual transmission in 70 to 80% of cases. So HIV can be transmitted from one individual to another during sexual or unprotected sexual intercourse. Also through blood transfusion, or in the drug addicts who inject heroin intravenously. And also HIV can be transmitted from the mother to the fetus via the placenta, which is a barrier that separates the maternal blood and the fetal blood. But in some situations or some cases, the placenta may transfer some viruses, some drugs from the mother to the fetus. And also HIV can be transmitted during childbirth and breastfeeding. It can pass through the maternal milk. So these are the ways of transmission of HIV: through sexual contact, blood transfusion, and from an infected mother to her fetus. These cases are the cases or situation that do not transmit AIDS: handshaking, restaurant, WC, showers, pool, pets, for example, hospital visit, school, public transport, public phone, mosquito, etc. So these are situations where AIDS is not transmitted. So you have to study these three modes of transmission of HIV. In the next videos, we are going to explain the cycle of HIV, how it multiplies within the host cell, and the progression of the HIV infection, as well as the possible therapies or treatments for HIV. Thanks for listening.